So I think we should perhaps start, actually. I'd like to start by thanking you for coming. Thank you for the speakers and also the moderators. My name is Giles Palmer, and it's a pleasure to introduce this subject of space biomedicine. For thousands of years, humans have adapted and learned to change their environments to suit ourselves, and in recent years, even control our reproduction. But as aspirations turn to the stars, can we really conquer this environment? I only recently discovered this sector of reproductive science, but investigations have been going on for decades. Indeed, since the year 2000, we've had people constantly in space. Now, there's a planned trip to Mars and there's focus on space tourism. These have made things more viable over the years. But what about the reproductive hazards of space travel for women and men going into space? Reproductive scientists and clinicians are looking at how space flight affects human reproductive health. And it's here that we're going to speak about this with reduced gravity conditions and high cosmic radiation, which they endure. So this work is vitally important to the space industry, but also for the future of space travel and also insights to more terrestrial applications as well. So we'll keep the bios short this evening, and all the full bios you'll find on the website, www.ivfmeeting.com. But please place your questions in the question section and enjoy a fantastic session. So over to you, Virginia.
Thank you, Giles. My name is Virginia Wotring, and I would like to introduce Dr. Fathi Karouia. He is a senior research scientist at NASA and on the research faculty at CU Boulder. A space life sciences expert with over 2 decades of experience advancing biomedical research in microgravity, leading over 35 experiments aboard the ISS. He spearheads innovation in in-space manufacturing and commercialization, bridging science and entrepreneurship to benefit both space exploration and human health on Earth. And also, I'd like to extend a warm welcome to Dr. Christopher Mason. Dr. Mason is the World Quant Professor of Genomics and Computational Biomedicine at Weill Cornell Medicine, where he also directs the initiative for quantitative prediction and the Cornell Aerospace Medicine Biobank. With a background that spans a dual BS in genetics and biochemistry, a PhD in genetics from Yale, and a clinical genetics fellowship at Yale Medical School, his career reflects a deep commitment to both scientific innovation and ethical inquiry. He was also visiting fellow of genomics, ethics, and law at Yale Law School. Dr. Mason's work has led to over 350 peer-reviewed papers, 5 patents, diagnostic innovations, and the founding of multiple biotech companies and nonprofits. He also holds affiliate positions at the New York Genome Center, Yale Law School, and the Consortium for Space Genetics at Harvard Medical School. Dr. Mason is the founder of the Metasub Consortium and the author of The Next 500 Years, which I recommend to my students every year, and also The Age of Prediction.
Thank you so much. Pleasure to be here. So Virginia Wotring, thank you so much for joining. She has had her training in biochemistry and a PhD in physiology and pharmacology from St. Louis University, where she studied the actions of medications during spaceflight missions, which is becoming more and more relevant for all of our work in the field. She is a Chief Academic Officer and Professor at the International Space University in Strasbourg, France, where I often I lecture whenever I get a chance and love those classes. And she's held management positions such as Deputy Director and for the Chief Scientist of the NASA-funded Translational Research Institute for Space Health. And she now does a lot of work in the science in the institute and managing it and deploying it. And so she has ongoing studies to look at the effects of the spaceflight environment on physiology, and especially those that may change how medications work, which again is more and more relevant as more and more people fly, taking many more drugs. So thank you very much, Professor Wotring. And then I think...
Thanks, Chris. And I guess without further ado, I would like to introduce the first speaker. So it is Dr. Begum Mathyk. She's an obstetrician and gynecologist with expertise in reproductive endocrinology and space medicine. She's currently completing a fellowship at the University of South Florida. She has held many different academic positions in Turkey as well as the US, and she currently serves as the associate co-chair for the Society for Reproductive Endocrinology and Infertility. And as a trained physician, she led the first gynecologist ultrasound in wetness and contributes to multiple international space health initiatives. Her work bridges clinical care and space biology, advancing innovation in women's health. Begum, floor is yours. Please take it away.
Thank you so much, Fathi, for this kind introduction, and I'm very excited to be here. Okay, and everyone can see my slides?
Yep, looks great.
Perfect, perfect. So this is my passion as an obstetrician and gynecologist, and I've been doing like over a decade research now in reproductive health. And now, you know, my interest is also involving space in the last few years too. Here's my disclosures. I'm currently at USF, but I'm also visiting scientist at Yale. We have collaborations going on there, and I'm currently leading the NASA's Open Science Data Repository, Female Reproduction, AWG, IAAS, BioAstra, and recently I just got back from Berlin for the Space Medicine Master's Program, and we are associated partner with them. So thank you for all these partners and collaborations. So first of all, this is the topic today is female reproductive health in space. And I will mostly talk about the oocytes, the hormones and embryos. But we need to understand that when we talk about the female reproductive health, it is more than a pregnancy or oocyte and embryo. So we need to understand the hormone synthesis, the regulations, the menstrual health, irregular cycles, abnormal uterine bleeding, the hormone synthesis in the ovaries, the follicle development, and the oocyte maturation in it. And of course, fertility is today's main topic, but we need to emphasize the congenital abnormalities like the Mullerian abnormalities. And what if pregnancy happens? This is one of the most questions I always get it. And is this an intended pregnancy? Are we able to get pregnant spontaneously in space? If not, what will be the IVF options for us? And what happens if we have unintended pregnancy, the medications like or spontaneous resolution of it, or do we need a surgical intervention? And if we get pregnant, are we going to face with the similar complications what we are facing on Earth? For example, preterm birth, miscarriage, preeclampsia, diabetes, gestational diabetes, or like other complications, or placenta invasion abnormalities. Another important part is family planning, infections, STIs, common gynecological issues. I work with fibroids, like fibroids are very common in females, endometriosis as well, the cancer risk, preventive care, the vaccination. Like my mind is always full of like how we will deliver the healthcare, maintain the reproductive health for women in their future endeavor in space, especially deep space. And also diagnosis, treatment, medical and surgical. And on the right side, I love these figures because it's not only about we ovulate every month, we have the menstrual cycle. It's very complex indeed. It always starts from the brain, the hypothalamus, the pituitary, and the ovaries. This is a very complex axis, and there's so many feedback loops. And then inside of the ovary, you see here, there's the folliculogenesis, and inside of it, the oocyte development goes. And it is so magical, our system, the oocyte development and everything indeed starts in utero when we were baby inside of the mother's womb. So this is very complex and very intricate system, and it's very fascinating and magical. And it's not only the oocyte is developing, the surrounding cells in it, the theca cells and the granulosa cells. So there is a lot of cellular communications. The reason I'm emphasizing this a lot because we will talk about the microgravity or gravitational forces as well. So all overall, female reproductive health, yes, I know I will focus on the oocytes and reproduction, but we need to make sure that it is all physical, mental, social well-being related to reproductive system throughout a woman's life, including reproductive age, menopause, and perimenopausal transition. And I would like to acknowledge always women in space, from the first woman in space, the first US woman in space, Cellulite. Her documentary is coming up soon. I'm going to definitely watch it. And we're going to continue our space journey as well. We're also entering a new era with the commercial space flights and like making space open to everybody. So I heartfelt congratulations and extend my gratitude for all the women in STEM and in space industry as well. When we look at these numbers, you can see it. The circles are flight duration, but look at the presence of women in space. I know the numbers are small now. It's almost around like 10 to 12% of the space flown human are representative of women. But still, it doesn't mean that we need to understand and investigate it. So we need to still put a lot of emphasis understanding the woman's health, reproductive health, and especially sex differences. And why space? That's a lot of question I get it from my colleagues. You know, you do already research on Earth, and why space? Because space is not only fun, it's innovation, it's exploration. There's a lot of economic growth in space industry, but you push the boundaries for science and technology. It is fascinating. You have to come up with ideas. You have to develop countermeasures. And you need to be good about troubleshooting and understand the system. It prepares us for Mars and beyond and improves life on Earth through research. Like there are so many examples of it, the organic development, the drug technology and everything. So I do love space, and there are many reasons that why we do space research. How about the space environment? This is another question I always get. Why do you do space research? Because I try to understand a woman and their body and the physiology, how it changed in an extreme environment and different environment. It's the same subject you're working on, but you really need to understand what will be the physiological changes in a different environment. And this is coming from the abbreviation of NASA, REACH. So we all know that those are the health hazards to human, radiation, gravitational changes. It's a hostile environment. You are far from Earth, confinement and distance and everything. And each of them are separately associated with health hazards like cancer, cardiovascular deconditioning, neurovascular deconditioning, immune deficiency, circadian rhythm changes. But these things can be isolated, associated, or you can have, most of the time, you have a combination of it. And our whole goal is to make human life safe there, reproduce safe, and becoming like an interplanetary species like we are doing on Earth. There are several platforms that you can do space research also. It's not always about like sending samples to space or, you know, examining and investigation astronaut health, but also there are conditions that you can mimic the space environment. So one of the biggest issues is radiation. We know that radiation has a direct DNA damage effect and indirect DNA damage effect through reactive oxygen species. And the direct DNA damage effect can be single-strand breaks or double-strand breaks. One of the things important about radiation is the age of the subject, the radiation dose and type. And we all know that human oocytes are sensitive to radiation, but also the oocytes from different species have a different sensitivity to radiation. This is on Earth data, like 2 gray dose can destroy 50% of the primordial follicles, which is very important. And these data usually, some papers says 2 gray, some papers says 4. Those are coming from 1989 and 2001. But we also have another data that like when the patients have cranial radiotherapy in their childhood, it may result in precox puberty and some pubertal or developmental abnormalities. And of course, we try to protect ovaries, like when there's in the radiation field. It's associated, like if you lose your follicles in the ovary, eventually you will develop the premature ovarian insufficiency. And the uterus is important. I know we always focus on ovaries, the ovarian aging, the impact of radiation on ovary, but also we need to think about the whole reproductive system, the fallopian tube, the uterus, cervix, and the vagina and the vulva. So what happens to the uterus is, if you expose to radiotherapy during childhood, there are studies showing that you have a reduced uterine volume, pregnancy complications associated, like preterm birth. What happens if you're an embryo or during the development, if you're exposed to radiation? The teratogenesis differs based on the gestational age. Usually in the first trimester, it's all or none, rule applies, and it depends on the radiation dose during the organogenesis period. So what do we know about space research? There has been ground radiation data, and it depends on the particles. So basically, there are two main studies. One of them is with the oxygen ions, heavy ions, which of course, like when you irradiate the ovaries, we eventually develop the premature ovarian insufficiency. It's not exactly early menopause, but you will have menopausal symptoms and the hormonal panel too. And eventually they develop, like, this is the mice, DNA double strand breaks, oxidative lipid damage, like there's so many disruption in the menstrual cycle as well. But the interesting is that those two big papers, there is differences in particles. Even with the same dose, it depends on the particle when you irradiate it, iron and heavy oxygen, depending on the decrease in the primordial follicle fold. So again, I want to mention that the age is important, radiation type is important, low red, high red, cosmic radiation, and the type of the radiation is important. And it could be detrimental depending on the dose. Like one of the studies with the 50 centigrade, in 8 weeks, you will lose 99% of your follicle pool. So radiation is one of the biggest issues for us. How about the gravity? So we all know, have a plenty of data from different organs coming, like simulated microgravity can change the aorta stiffness, the arterial intimal thickness, and everything. We have also data like the hypergravity, like when the pregnant rodents exposed to hypergravity, the pups had increased mortality and decreased birth weight, and also placental weight impacted. And as far as I'm aware of, if you go about 4G, like there wasn't even like live pup birth. So we need to understand again, it's not only microgravity, but also the hypergravity and different gravitational forces impact on our body. How about the cellular level, about the microgravity? We evolve on Earth, 1G, and when you look at our human body, we are like majority of ourselves like made up of molecules, the water, and then there's a lot of cellular interaction at both the molecular level, subcellular, and organ level. We have plenty of data that microgravity, whether it's a real or simulated microgravity, affects the mitochondria's function, microtubule function, and cellular cytoskeleton. The reason I'm emphasizing is during the oocyte development and during all these follicular genesis and everything, but especially with the oocyte development, you have meiosis and mitosis and a lot of spindle division. So this is very important, like the ultrastructural impact of micrograviton cells. Another perspective is circadian clock. We have circadian rhythm, in the central nervous system, but there are clock genes exist in reproductive organs, which has not been a lot exposed and explored in the space era. But when you look at ISS, it orbits the Earth every 90 minutes. So it's just like an amazing environment, and each planet, this will be the different. So while we are understanding the radiation and gravity, we need to be mindful about the circadian clock. We have data on Earth on circadian clock, basically shift workers, they may develop insulin resistance or some other issues as well. So when we focused on reproduction, there has been plenty of data. So is it something new? No, it's not something new indeed. The first, one of the first studies coming from 1966, there are plenty of different kind of animal studies, like from shuttle mission time, Gemini, one of the first in 1966, the frog embryos reproduce, the sea urchin, and one of the first vertebrate is the Mathyk fish. You can see it in this figure, lay eggs and hatch. And an interesting one is the cockroach. It was a Russian mission in 90s, and they just trace back to the grandkid, I said. So they were looking at the morphological differences. But my main focus is the more rodents and especially human. So we're going to dive more detail in that. I would like to start with the hormones. So we had a great paper package collection last year. It was released, and one of my main question was the hormonal changes. I couldn't have access to the actual hormone levels, but also I was interested to looking at the gene expression profile changes related to insulin and estrogen. Then also we decided to make a big review about like what happens. Let's divide it up all the female reproductive system and see what are the data, anything about rodent and human coming. And these are some of the papers coming from this space paper package. So I will just touch base briefly. So why estrogen? Estrogen is also, of course, we mostly know it as a reproductive hormone, but indeed estrogen is very closely related to metabolism, and the receptors are expressed almost, various tissues. It's important for bone health. It's important for cardiovascular disease, neurocognition, muscle health. So that was one of the reasons that I focused on the estrogen-linked genes. And it's not only in females, you can see in the males as well. It has various receptors, and there is both genomic and non-genomic signaling. And also when we talk about estrogen, we have different forms of it. The main focus is, of course, in the reproductive age woman is the estradiol E2. Another one was the insulin. The reason is that insulin most of the time known for metabolism and glucose, lipid, and protein, but also in reproductive tract, insulin has receptors. So that's what I would like to focus, and there is a smooth interaction between estrogen and insulin because estrogen enhances insulin sensitivity, and we have the clinical data backing this up. And this is again widely expressed in many tissues and also in both sexes. So we used open science data in this one. Thanks to all collaborators, Fathi, Chris, Jackson, Fumi, everybody. And we had plenty of data from both rodent and human. We look at the estrogen and insulin linked genes, how these gene sets change in space-flown samples. When we look at the female mice, I couldn't have the ovaries way back then, but we try to understand that from the same mice, what happens in different tissues. And it's not so much surprisingly, but the liver was the most impacted organ. Why this is important? Because liver is main hub for most of the drug metabolism. So I think it's very important to understand how the liver metabolism changes. And the number of the genes, of course, was differentially expressed genes was the top on the liver. And then we focused on the human data. This is from the plasma cell-free RNA. I have different heat maps, but I cannot show all of them due to time limit, but I want to focus on the estrogen signaling linked genes. Those are all genes before flight, after flight, and they are upregulated and downregulated. The interesting thing when I look at this heat map is genes can change post-flight. Some of them persist in longer time, but for some of them shorter time, upregulated then downregulated. And some of them are related to hormone metabolism. One was leptin here, one was oxytocin, and this enzyme is important for this is the gene synthesizing enzyme important for steroidogenesis, cortisol, and other steroid hormones. And this is a clock gene as well, and this is the estrogen receptor 1, one of the main estrogen receptor gene. So it's very important to understand. And when we look at these gene changes, they were associated with immune pathways here, but also some metabolic pathways. And then after this paper, we wanted to focus on what is the data out there, the rodent and human data, looking at each different perspective or each section of the female reproductive tract, starting from the HPG, the menstrual cycle, steroidogenesis, oocytes, aging, and uterus. I would like to also emphasize and focus on epigenetics and the Dohat hypothesis. So basically what we expose inside of the mother's womb or the uterus, it may have a consequence effects after the birth. This is very important to understand. And one of the common questions I'm always getting at, is there a risk for ectopic pregnancy? I don't know, because we don't have the data how the fallopian tubes behave in longer term microgravity or when they expose to radiation. So this is almost 20 pages. I cannot go in detail, but if you're interested, you can take a look at them. And this paper is only focusing on rodent and human data, but I will touch base the oocytes and the embryo part. Another thing we were considering, what happens if you cannot get pregnant in space, if you need an IVF treatment. The IVF on Earth now evolved so much, since the first IVF baby, and the lab equipments and everything, and our pregnancy rate as well. But the other question is that the germ cell quality and quantity will be the same. How about the current technologies that we use, like in vitro maturation, or the culturing of the embryos and the oocytes, or the freezing methods, cryopreservation? Can we use the same thing in space? So basically preserving the genetic diversity, it's very important. It's not only for human. When you look at the literature, people are trying to preserve the seeds for nutrition or any kind of animal genetic component for the future. And yes, we are going to mention and discuss there are some challenges currently if you want to apply all the IVF tools in space environment. So I think this is one of the things people are interested in to learn more about what happens to our oocytes and embryos. So my focus is that, as I mentioned, like most of the rodent studies and human, if it's available, there's plenty of data because of the microgravity and some partial radiation. The oocytes morphologically and also at the cellular level impacted by the both spaceflight and simulated microgravity. And you can see it here, like the decreased rate of oocyte maturation, disrupted meiotic spindle organization. When I mentioned in prior slides why the microgravity's impact is important for cytoskeleton and the tubulin structure, this is why it is important. And the other data is like they show that decreased oocyte maturation, GDF9 expressions, especially plenty of data about the abnormal structure, like the impacted mitochondria, lipid droplets, and etc. One question is that what about the hormone synthesis? So this is from 2021 space flown mice. Surprisingly, and the authors mentioned that they look at the ovarian tissue, all the enzymes synthesize steroid hormones, and there was no significant difference. Although it looks like this is the flight group, it looks like decrease, but it wasn't statistically significant. And here you can see the estrogen receptor and GDF9. So there wasn't a statistical significance. And they also mentioned that it may be feasible for rodents to menstruate like continue their cyclic activity. And also I was looking at the granulosa cells because it's very important for the estradiol synthesis and everything, TECA cells, granulosa cells. So I started to look at literature in detail. And of course, like simulated microgravity has unfavorable effect on the porcine granulosa cells, and it affects the cell cycle and definitely changed the morphology. And another study follow-up is looking at the mice ones, showing that like the maturation is impacted, again, unfavorable, and the cytoskeletal organization impacted. One of the interesting studies is that, how about the human oocytes? This study, there is no transcriptomics data, it's only morphological, but when you expose human oocyte to microgravity, you can see it here, the perivitelline space is expanded, and then the zona pellucida becomes thicker, and there are other morphological abnormalities about mitochondria. So unfortunately, at this level, with the current knowledge we have, microgravity has been shown that causing structural abnormalities in mice, mostly rodent oocytes. And we decided to look at the impact of hypergravity on human oocytes. We were expecting something like unfavorable, but surprisingly, we had this protocol. We exposed the control versus hypergravity. We noticed that time of those were the GV oocyte siblings, and this was a time-lapse monitored. It was shorter when the human oocytes exposed to hypergravity to approach the maturation process. So this is our ongoing study still. We try to understand, or we did some look at the gene expressions, and there is some underlying unfolded protein response, but we are still investigating the importance of this data. So when we are moving to embryos and organogenesis, it's not only fertilization, like it's so magical. You have the sperm and egg, fertilization happens, two-cell stage, morula, and the blastocyst. So embryogenesis is very important, but we need to understand also in utero organogenesis. Like even if you assume that in space environment you get pregnant, like your embryo implanted to uterus, that embryo needs to continue to develop, and then the organogenesis needs to happen. And then you have the fetal development parts, first, second, and third trimester. And even after that, let's assume you deliver in space, there should be the postnatal development, especially for brain. And with the embryo, when the blastocyst happens, this implant to the uterus, the blastocyst has two important layers, the inner cell mass, which is going to develop the embryo, embryoblast, and the trophoectoderm. This is important for placentation. And you can see this is like very magical for me. Like since I was in medical school, the human embryogenesis and organogenesis, it's just fascinating. So what is the data about embryo development for space? Most of the time, mouse embryos sent or studied in two-cell stage because they want to understand that the development from two-cell to blastocyst. And in early studies like 1990s, early 2000s, it may be due to the experiment conditions. The mouse embryos did not develop blastocyst, unfortunately. But one of the important groundbreaking study is coming from 2016 to 2020 in an orbit mission. And why I'm saying it is groundbreaking, because this is from China and they developed an embryo culture incubator similar to what we have in the IVF lab, because they wanted to also monitor and look at the images, how the embryos develop. Still, there is a success. The two-cell embryos develop blastocyst, but it was still reduced rate when you compare to Earth, like their ground controls, so 34 to 60%. There were other issues because you need to look at the cellular differentiation, like we mentioned, that the inner cell mass, the trophoblast and everything. The cellular differentiation was affected, but also there was an increased DNA damage in the orbit float ones. And they also conducted a ground radiation study, tried to understand the impact of radiation on those embryo development. And again, depending on the dose of the low lead, this is a low lead radiation, exposed 64 hours. Depending on the dose, the blastocyst development was decreasing. Another fascinating study coming from Japan. So in 2022, they published, because they tried to develop a device, they wanted to use the high osmolarity vitrification and they want to keep the frozen embryos at -80. So there are some differences. Currently in IVF labs, the embryos are frozen more like, like in the cryo tanks, it's minus 196 Celsius. And this is important because of the crystallization of the embryos and oocytes. So we need lower temperatures than the -80. But they try to troubleshoot working on the frozen mouse embryos, to understand that can we change the cryoprotectant? Can we do high osmolarity vitrification? And the second thing is that they wanted to develop an automated system. Not every astronaut is going to be an expert like seasoned embryologist. So they wanted to make it feasible, usable for the astronaut, like, so they can change the culture medium. That was a good thing, but this is like a lot of like resilience, trying different methods and everything. Still, when they told the embryos, some of them reach out to blastocysts, but this just depends on the culture environment and the device that they use. So yes, the mice embryos can develop in space, but the rate is still lower than on Earth. And this study was followed by their space embryo project. So the mouse two-cell embryo sent to ISS in a frozen state. So the ISS is going to be the microgravity, and there is an artificial 1G facility in ISS. They were taught and cultured there for 4 days. And there is, of course, always the ground control. But the interesting thing, I really congratulate all the authors and the team, because this equipment, like doing a self-sustained small IVF unit, the equipment development takes years, 2012 to 2021, to make this experiment happen. And they look at the blastocyst rate, the numbers of differentiation, DNA damage, and next generation sequencing analysis, RNA sequencing. This paper, I was very curious to see the haploidy, aneuploidy rate, but it wasn't mentioned there. There are some differences also. They use fixation for 0.99% PFA. Usually it's 4%. So there are some differences. Still, we want to mimic the IVF on Earth and try to make automated systems, but it's not exactly the same what we are doing now. So what happened? We sent those two-cell embryos there. We taught them in this like our new device, and like they were looking at the blastocyst rate. So those are the, first of all, not all the embryos survived. Those are the ones survived. This is the retreat number ones. And with the Earth, this is your control. So 61% reach the blastocyst, while the space one, 23% reach the blastocyst. And this is still space. You have the artificial gravity station, and 31% reach to there. So basically, yes, mouse embryos developed in space, but still the rate is lower than compared to Earth. This study, compared to prior one, the orbit mission, they didn't see significant difference in the inner cell mass and trophoectoderm differentiation. But I think the number is less also here. They just analyzed the 5. And they also look at the DNA damage, and there wasn't non-significant differences. So it is promising, but still there is a lot of troubleshooting with the correct equipment and independent culture media change as well. So this is the quick summary of the basic science, like the oocytes and embryos. I would like to mention a little bit clinical research because it's also pertaining to reproductive health. One of the main things like we need to understand is the cancer risk. This is currently, we have an ongoing project in NASA Female Reproductive Analysis Working Group about the breast cancer risk. Why breast cancer? Because it's common in women, like 1 in 8, 1 in 10. It increases with age, and ionizing radiation is a known factor. And history of ionizing radiation exposure to chest is a known factor. There is also hereditary risk factors as well. So we look at the mammary gland that was space flown from female mice, and there's a difference depending on the age of the mice. So the gene expressions in the mammary gland. So this is still ongoing, but I think we really need to emphasize the gynecological cancers because there is the oncofertility component of it. And we need to understand like how the cancer risk increase. If so, how we are going to do the oncofertility treatments for space. Another thing I was working on using of the gynecological ultrasound, and I would like to test it during a parabolic zero-G flight. And why this is important, again, everything links to the knowledge that we know on Earth. Gynecological emergencies are common, abdominal pain, and ultrasound is our primary tool to understand the differential diagnosis. And anything, either it's a bad or good thing underlying, that can also impact your reproductive health. The other one was the IUD placement on a model. Shauna, Kelly, and Nora, they were working on it. And I was also focusing on the microbiome collection, try to understand how the vaginal microbiome changes before and after flight. And why vaginal microbiome? Again, it's related to reproductive health because we have plenty of data coming from Earth, like with reproductive health diseases like endometriosis or in other condition or cancer, the microbiome can change. So this is the gynecological imaging. It is not only for diagnosis, but also for monitoring. It's important. And also you can do experiments. You can understand the blood flow. And again, why this is important? Because we talk about the microgravity, the cephalid fluid shift. So how about the lower extremities? And uterus and ovaries are highly vascularized organs, and they have a direct blood supply from big vessels like abdominal aorta and iliac arteries. So this is also, you can understand the physiological processes, how it changes for space. And this is again unpublished data. We are still analyzing it. I was surprised to see that there was a more robust change in vaginal microbiome than the oral microbiome post-flight. And currently, there's another thing that you can do it if you want to do space research is the isolation studies. Our proposal got accepted last year, and then we will work on how the isolation affect like the menstrual health and reproductive health, basically. This is the next upcoming project. It's very important to talk about this topic. It's raising awareness. So this is what I've been trying to do it. And I love to collaborate. We have a very great group that everybody collaborates in AWGs and other societies. We should have to push the boundaries and try to understand. We definitely need more data about the female reproductive health, including the hormones, the oocytes, the embryos, and everything that we talk and we haven't covered here so far. And what is the future direction? Definitely hormone sensitive and hormone responsive tissues. We need to understand how the hormones act. The gynecological cancer risk, the gametes are important. We still don't know about the human gametes, how it responds for both radiation and different gravitational forces. And then the more clinical thing, like, remote diagnosis, monitoring, targeted delivery systems, and imaging. So we need to continue for both clinical research and translational science. One of the things I try to do my best to get more information with our collaborators to standardize the protocols, because every data is so valuable coming from space. It's expensive, difficult to replicate, almost impossible. So we have to have standardization protocols and then sharing the data among others. And of course, biotech and like technological development is important. I just want to say thank you to everybody, like all my friends, family, mentors, mentees, collaborators. This is a great field to be in, and we welcome everybody. And thank you. Then this was from my aerospace medicine clerkship rotation in Johnson Space Center. Was one of the best moments.
Dr. Mathyk, this is an amazing story. Thank you so much for the comprehensive overview of the female reproductive system in space. We'll press ahead here and hear the complement to that story. I'd like to introduce to you two experts who will speak on the topic of male fertility, Dr. Blair T. Stocks and Dr. Jeffrey Jones. Blair T. Stocks, MD, PhD, who is an assistant professor at the Division of Male Reproductive Medicine and Surgery within the Scott Department of Urology at Baylor College of Medicine. Blair's interest is male infertility and men's health, and complementing his clinical practice, he runs an active basic science laboratory focused on mechanisms of male infertility. I'd also like to introduce Dr. Jeff Jones, distinguished professor at Baylor College of Medicine, a former colleague of mine there, and the executive for inpatient services at the Michael E. DeBakey VA Medical Center. Jeff has an extensive background in medicine, aerospace, and urology. He has held pivotal roles, including chief of urology and NASA flight surgeon for shuttle and ISS missions. He is a diplomat of the American Boards of Urology and Preventive Medicine. He's contributed significantly to research, education, and clinical advancements, earning numerous accolades such as the H.G. Stevenson Award and NASA Group Achievement Awards. He's a prolific author and innovator with patents in prostate cancer detection and radiation exposure mitigation. His expertise spans space medicine, urology, and preventative health, making him a leading voice in his field. Jeff, over to you.
Well, thank you very much, Virginia, and welcome to all of the spaceflight enthusiasts. I'm going to tee up Dr. Stocks. He's going to talk about the male reproductive biology and talk a little bit about what Dr. Mathyk had already introduced, which is the radiation environment. Neither Dr. Stocks or I have any disclosures that will be of conflict of interest in this particular discussion. However, if any of you have any interesting offers, we'd be happy to consider that and induce a conflict of interest. All right, so Dr. Mathyk talked about the space environment, and just to reiterate, we have 5 key risks to human health: the space vacuum, microgravity, temperature variations, extremes in temperature, the potential for micrometeoroids and debris in space, and then the radiation she mentions, the charged particles and other types of space radiation. Space environment, now we're talking about microgravity, and we know that a number of the microgravity effects include all of the human physiologic systems, and these systems will adapt to microgravity over time. And a number of the molecular changes that happen during microgravity can either be synergistic or antagonistic with other space environmental effects, such as radiation. So there are 3 principal sources of space radiation: the geomagnetically trapped radiation, the solar radiation, and galactic cosmic radiation. We'll talk briefly about each. In terms of the radiation that's trapped in the geomagnetosphere of the Earth, we divide it into the inner and outer Van Allen belt. And most of the exposure while in low Earth orbit will be from this trapped radiation. There is an area in the South Atlantic that dips down lower in altitude, and we call that the South Atlantic anomaly. The solar radiation from our sun is fairly constant, what we call the solar wind. It varies in an 11-year solar cycle from what we call solar minimum to solar maximum. During that time, there may be slight changes in the number of low energy protons that come from the sun, but the main risk in solar max comes from the likelihood of solar particle events. And those are typically flares or coronal mass ejections, and they can produce both high flux and high fluence of solar protons, and some of these can be high energy and therefore be more dangerous to the crew. Now, the galactic cosmic rays come typically from supernova and distant quasars. They're isotropic, they're ubiquitous throughout the universe. The difference with galactic cosmic radiation as opposed to solar radiation is instead of just these hydrogen and helium ions, you can have iron and other heavy ions that come at relativistic speeds and therefore have a much higher lineal energy transfer. And that can cause more risk to the human. And we measure radiation based on how high or low the linear energy transfer is and what the relative biologic effect is. So we have both the passive dose in gray and the biologic dose in Sievert. And we actually have weighting factors depending on the type of radiation. So X-rays and gamma rays are a weighting factor of 1, whereas some of these higher energy particles can have weighting factors that vary from 5 to 10 to even 20, especially high energy neutrons that are a result of collisions between the high LET particles and shielding. And these higher weighting factors can produce more tissue damage. Now, here are radiation limits, and you can see that the radiation limits for astronauts in space are 10 times that for terrestrial workers. So terrestrial limit is annually 5 rem for someone in a nuclear reactor. It's 50 rem per year for an astronaut. And the limits that we have for 30-day exposure are based on the effects of testicular fertility, which at this level of exposure in 1 month is reversible. So that's how we define that lower level of limit for astronauts. Now, what are the exposures like in space? And you can see on Earth, living at sea level, you get about 1 millisievert per year. In Denver, at Mile High City, 2 millisieverts per year. Or if you go on ISS for a 6-month mission, you can expect to be somewhere between 160 and 200 millisieverts. If you go into interplanetary space, the rate is much higher, where we can expect somewhere about 1.8 millisieverts per day. And on the Martian surface, about 0.8 per day. So the total amount for a 3-year Mars mission with 6 months outbound and return would be somewhere between 0.7 and 1 sievert. So that's a significant increase compared to what you would have on Earth. And you could talk orders of magnitude here. This would be living at sea level. This would be getting an abdominal CT scan. And then this would be a 180-day mission transit to Mars with a 500-day surface stay. You can see that's 2 orders of magnitude higher. As Dr. Mathyk mentioned, there are the potential with radiation of having either single or double strand breaks. The double strand breaks are much more likely to produce mutations that could lead either to cell death or to oncogenesis. And as mentioned earlier, the low LET radiation is more likely to produce a single strand break, whereas the high LET radiation is more likely to produce a double strand break. And in addition to injury to the nucleus and the genetic material, there's also the potential, as mentioned earlier, to produce reactive oxygen species that can interact with the cellular organelles and potentially damage membranes or other parts of the cellular structure. Now, the radiation effects can be divided between acute and chronic. The acute effects can occur from a large solar particle event, as occurred during the Apollo missions between 16 and 17, where we had a very large solar proton expression in 1972. Most likely from this exposure, the crew would have a prodromal syndrome, but there is the potential, if a very severe SPE in interplanetary space, you could have acute radiation syndrome. And so that highlights the need for a storm shelter. Now, the chronic radiation effects are more likely to be due to this prolonged exposure from the GCR and occurs over many years. It can produce malignancies, it can produce damage to the central nervous system, and can produce degenerative effects. And these degenerative effects can include things like cataracts or clouding of the cornea. It can include vascular effects like fibrosis and cardiomyopathy, or it can produce effects to the endocrine system, as mentioned earlier. And the endocrine and exocrine glandular organs can be affected from the radiation that the crew receives during a space mission. Now I'm going to hand off to Dr. Stocks.
All right. Thank you, Jeff, for that wonderful introduction. Again, I'm Dr. Stocks. I'm one of the male infertility specialists here at Baylor, and very excited to be giving a little bit of this talk and get my feet wet when it comes to male fertility in space. So just as a background, similar to the last talk, just wanted to give a brief review of male fertility. So as we all know, it starts in the hypothalamus, where your hypothalamus releases gonadotropin, releasing hormone, which then talks to the anterior pituitary to make two very important hormones, one of which is luteinizing hormone, which stimulates testosterone production through Leydig cells, as well as follicle-stimulating hormone, which stimulates Sertoli cells, or the nurse cells, that promote spermatogenesis. And this testosterone itself within the testicle, or intratesticular testosterone, is very important for the maturation of sperm. Just to go over spermatogenesis briefly, it's a very complex process as well, similar to females, in which there's many coordinated steps to actually go from that primordial germ cell to spermatogonium, spermatocytes, and then finally spermatids and mature sperm. And it is important to note that this process does take about 74 days to go from germ cell all the way to mature sperm. So if we're thinking about prolonged space flight, the effects of radiation, gravitation, can also, affect this very delicate process. So proposed space factors affecting male fertility. Thanks, Dr. Jones, for a great background, but first and foremost is going to be exposure to ionizing radiation. Specifically, both the type and the duration of this ionizing radiation is important. So again, space flight members can experience galactic cosmic ray radiation of high energy particles, solar particle events, as well as the trapped radiation such as that experienced in the Van Allen belt. Another factor, too, of course, is alteration in gravitational forces. We know that astronauts and crew members experience both events of hypo and hypergravity, and there is a good amount of basic science data that showing that these gravitational force changes can affect the HPG axis, leading to, in some cases, decreased testosterone production and testicular size. And then finally, it's just an important component to mention is just the age requirements of astronaut training and execution. With more and more astronauts not being of the Mercury and Apollo era, we are having older male astronauts join the force. As we know, too, that just age alone, outside of the factors of ionizing radiation and gravitational forces, does affect semen quality and semen parameters. So we know that volume decreases with age, our motility, morphology decreases, increases of DNA fragmentation can be seen, as well as gradual declines in testosterone levels. As we all know, that testosterone decreases with age. So I kind of wanted to split it up to two events or two kind of types of space missions that we've seen so far. So deep radiation exposure is generally thought to be above Earth's magnetosphere, and thus far, the Apollo missions running from 8 to 17, 1968 to 1972, there were a total of 24 astronauts, all male, who experienced some degree of deep space radiation exposure. Just like Dr. Jones mentioned, a deep space average exposure is about 1.8 millisieverts a day. Comparing this to low Earth orbit, it's about a third of the deep space exposure, and then terrestrial exposure is about 100th of that. But this is very important to think about when we're thinking about prolonged lunar and Mars missions. On the surface of the moon, about a millisievert a day, whereas the surface of Mars is predicted to be about 0.7 or 0.8 millisieverts a day. And then in addition, if you think about the potential of prolonged 6-month travel duration to Mars, this is obviously going to compound. So just out of interest, I thought it was interesting to look at the 24 Apollo astronauts and just see from a fertility perspective what their fathering rates were. And it was interesting, of the 24, 22 are actually fathers, the majority of which had their children prior to their space missions, but at least there are 2 instances, both William Anders as well as Edgar Mitchell, who actually fathered children after their space flight, at least with a small data point suggesting that 2 males exposed to deep space radiation were still fertile after their missions. Interestingly, if you think about the amount of radiation they received, it was actually fairly low because their flight durations were short. They were about 6 to 9 days, respectively, and of the radiation recorded, it was only 1.6 or 1.4 millisieverts, which is well below the occupational limit published by NASA at this point. But moving more toward the current era, low Earth orbit radiation exposure, so this is below our magnetosphere. Generally, the space shuttle missions running for about 30 years from 81 to 2011, 135 missions with a total of 355 astronauts, as well as the ISS missions more recently. There's been about 129 with about 269 individuals participating. But this is interesting more to the compounding effect. So yes, low Earth orbit radiation exposure is less, but it can be compounded by the duration of travel. So the longest ISS duration was about 370 days, exposing this cosmonaut to about 370 millisieverts. Unfortunately, no public data exists regarding fathering post-mission for either of these missions. But just to get to the point, astronauts are aging again. So if you look at the average age of the Apollo astronaut, 39 years, the average age of the space shuttle astronaut, about 41 to 47 years, and the ISS is 48 years. So also to the fact that astronauts are older as fathers compared to earlier time. What I think is kind of most interesting is what we can glean from animal models regarding male fertility in space. So there's a pretty interesting set of papers I'd just like to review briefly using simulated gravitational forces on the ISS. So it's basically a centrifuge for mice, which is pretty interesting, where they can house mice and then with the centrifugal forces actually recreate gravitational forces that are either similar to Earth at 1G or microgravity conditions such as thousands of that. And then interestingly, you can actually compare this to ground control mice as a good control. So I'd just like to sort of review what this literature has shown us thus far. The first one is basically a group of mice that went up on the ISS or are exposed to these conditions for 35 days. So again, looking at the groups of ground control mice, artificial or 1G gravity or microgravity. After 35 days in space, the animals are sacrificed, and you can see that the tissue weight of the testicles was similar between groups, unchanged. The second graph looking at different motility parameters between the groups, showing basically very little or no differences between the motility. And finally, getting to the point of, are these mice fertile? You can look at the birth weights of the F1 generation, finding no differences in the F1 generation birth weight, nor the actual offspring rates. So kind of taking away from this, is basically 35 days of exposure in low Earth orbit didn't really have any lasting effects when it came to fertility in these mice. Just kind of from a different lens, this is I believe the same group, but what they did was ask the question, if we cryopreserve sperm from mice and send it for a longer duration on the ISS, does that affect the ability of that sample to be affected in terms of IVF down the road? And what they did is basically controlled cryopreserved sperm from ground control mice versus those exposed to 9 months on the ISS. And which was interesting, they did find increases of DNA damage on these cryopreserved sperm, as to be expected due to the radiation exposure. But when you actually looked at the birth rate of the mice, they were actually similar between the groups, suggesting that any sort of DNA damage occurred could be overcome by the female oocyte itself. So take away more DNA damage of those experiencing 9 months of low Earth orbit radiation exposure, but no differences in the F1 generation. There was an interesting point I'm going to lead into my next point, was they actually bred mice from this F1 generation with each other and found that they didn't have any difficulties in breeding in F2 generation, but it does kind of bring into the question, are there lasting effects of radiation exposure that can be passed to further generations? from this, it kind of gets to Dr. Mathyk's point too, is epigenetic modifications. So these were two papers briefly that looked at changes in demethylases and deacetylases expression in mice testes tissue that was exposed to spaceflight. And basically in this first paper, after 24 days of spaceflight, they found decreased expression of a DNA methyltransferase, sorry, as well as decreased expression of a histone deacetylase, suggesting that even 24 days of spaceflight can affect epigenetics of the testicular tissue and sperm. And similarly, this other group did a similar experiment. I think it was 35 days or so, but basically they were able to find changes in transcription factors related to methylation patterns, differences in mRNA expression that could account for different genetic silencing, as actually well as increased expression of DNA replication genes in the F1 generation, suggesting that some of these epigenetic modification can be transferred to subsequent generations despite such short radiation exposure. Why epigenetics of male fertility is important. We know that age, obesity, diet, smoking and alcohol, radiation and chemotherapy can actually induce epigenetic disruptions. And why this is important is that epigenetics of sperm may contribute to differences in neurodevelopmental changes, metabolic health, cardiovascular health, as well as cancer risk. Currently in lab, we're actually using a pretty interesting semen analysis kit that is looking at epigenetic parameters of sperm. And these have been shown to predict different intrauterine insemination or IVF success outcomes. So really, we're just on the burgeoning field of understanding epigenetics of male fertility. So just sort of conclusions from my review of everything. It appears, at least from the data thus far, that shorter duration missions, especially at low Earth orbit, have lower radiation exposure overall, and it really doesn't appear to affect male fertility directly. However, there is emerging evidence demonstrating possible changes in the sperm epigenome secondary to spaceflight, but really we don't know what risks, or if any risks, do occur and what they pose to the F1 and F2 generations, and this remains to be determined. So the next two bullet points are just my personal viewpoints. I think, prior to any space flight, I always say, be that dad you are today. We don't know the changes, right? So maybe it's important to just cryopreserve prior to space flight to try to mitigate any of these risks. And then similarly, just figure out the dad you are today. So maybe we should be considering pre-flight assessment by a male fertility specialist prior to flight to see if there's other conditions that may be present even before space flight, such as some degree of oats, hormonal imbalances, or even anatomic etiologies such as a varicocele. So thank you for your time. I'm going to head it back to Dr. Jones real quick.
Okay, well, thank you, Dr. Stocks, for that great review of the male physiology and the risk. I'm just going to talk about, now that we know what the risks are with microgravity and radiation, what can we do about it? Well, we can certainly measure the radiation in space, and that's done commonly on space vehicles to allow us to understand the exposure of the crew. And then we need to do something to mitigate that exposure. And we can do that best by getting where we're going faster, spending less time in interplanetary space. So all you rocket engineers, get on it, get designing new vehicles to get us to where we want to go in the solar system a lot faster. If we can't get there faster, then we need to shield the crew during their transit. And the shields we have are pretty good for low LET radiation, but there's no shield that we have currently in space that will shield against this high LET radiation from the GCR. So that's going to get through and we're going to get exposed. So what can we do about that? And I'm going to talk about a few biological potential countermeasures. There are three types of potential biologic or pharmacologic countermeasures. We call those radiomodulators that act to increase the baseline resistance of the individual. And then we have radioprotectors that act at the cellular level to neutralize these reactive oxygen species we've been talking about. And then there's radiomitigators that act systemically to accelerate the post-radiation recovery and prevent any potential complications of that exposure and the downstream effects. And there's some examples of radiomodulators. We've been looking at natural compounds from a variety of food sources, and these can be given together in combination to create a formula. And these are especially notable is the radioprotectors, and this is one that has clinical applicability for patients getting radiation therapy. It's called imifostine, and it's given to people getting head and neck cancer treatment, and it's approved by the FDA, but it does have a few side effects, so it's not an ideal agent. It does seem to reduce some of the effects we have seen in terms of radiation mucositis in these patients. Then there's the radio mitigators, and this would be stored in the vehicle prior to the crew going to orbit, and it could include their own blood, their own stem cells. It can also include things like growth factors. We give Neupogen to stimulate white cell production, and we give erythropoietin for red cell production. So these would be available for the crew that had some type of a long duration exposure. And they can be very effective in reducing cellular injury and cellular damage. And this was a study we did looking at a chemo prevention cocktail and reducing the number of chromosomal aberrations. And we have even developed the ability using the manganese superoxide dismutase liposomes to reduce the likelihood of death after a lethal exposure in rodents. And so with these types of countermeasures, we could protect the crew and their reproductive health if it were a radiation exposure scenario. We've even tested them in astronauts in the nutribuoyancy laboratory and seen improvements in their ability to do work by reducing their fatigue and reducing their lipid peroxidation products. This is the combination therapeutic that we have flown on 2 space shuttle missions and 1 ISS mission and shown improvement in the markers of oxidative stress. So in conclusion of our talks today, it appears interplanetary radiation exposure is a significant biological threat to the crew and one of the biggest hazards for our future exploration class space missions. And the main hazard comes from a combination of low LET radiation from the solar source and then high LET radiation from galactic cosmic events. The potential to use pharmacological countermeasures could reduce both the biologic effects of acute exposure with the radiation protectors and mitigators, and then the potential for chronic exposure with our radiomodulators. And with that, we're going to conclude. Here's our references. Here's some things we'd like to do in the future. And turning it back over, going to stop sharing and to our moderators, Dr. Karouia or Dr. Wotring.
Great job. I think, and in this case, it comes back to me for this one. So Dr. Jones, Dr. Stocks, very beautiful work. Thank you for, it's good to see you also. It's good to say hello and really fascinating work. Actually, I'm excited for this symposium because we have a lot of really the key primary literature that's come up over the past 15, 20, even 30 years that we should build upon all this work. And so, yeah, I had one question, actually. I put a link in the chat about some single cell sperm sequencing that's now possible. I was curious about ways that you've thought about it to look at, sperm, individual separate sperm differences and like what's the proportion of sperm you think that could be affected by some of these radiation effects? Like, there's some theories about selective pressure for autism risk and curious about paternal age effect, kind of some of those questions and what you think.
Yeah, no, I think that's a fabulous question. I know with single cell transcriptomics, I think that'd be a great way to start answering this question, sorting out individual sperm and kind of running mRNA expression data, genetic data. I think that'd be an awesome place to start. And definitely, if we have availability to animal models, to start there. But I think that's, a fabulous question to start looking at that question specifically.
Yeah. And Chris, that sperm self-select, right? So it's only the fittest that make it to the egg and penetrate the zona pellucida. So if you have one that's defective because of heterogeneous, radiation damage, it's not going to be the guy making it all the way there.
Yeah, that's very important. Yeah, I was definitely, I think, the lost sperm, and I somehow made it. I don't know. I definitely was ideal. But that was thing, and I think we're a little bit long on time, so I might just do one quick question. But thank you very much, and really fabulous work. And I'm getting some messages to switch over now, so we're going to hop to the next speaker. Thank you again very much, Dr. Jones, Dr. Stocks, for their wonderful talk and great work. And now for having to jump into a talk on the ethical issues for space-based fertility and reproduction, please welcome Dr. Paul Root Wolpe, a renowned bioethicist whose work bridges science, medicine, and ethics with a special focus on the ethical frontiers of space exploration. Dr. Wolpe served as NASA's senior bioethicist for 17 years, advising the agency on the ethical implications of space research, human experimentation, and emerging biotechnologies. He's the Raymond F. Shinazi Distinguished Research Chair of Jewish Bioethics and Professor of Medicine, Pediatrics, Psychiatry, Neuroscience, and Sociology at Emory University. He is a key founder and pillar in the field of neuroethics. Dr. Wolpe is also a global leader in ethics related to AI, human enhancement, and genetics, and he led the Emory Center for Ethics for 16 years, co-chaired the APA's Ethics Commission, and founded the Being Summit to shape global bioethical standards. He also has a TED Talk that's been viewed over 1.5 million times, with extensive work in academia and public discourse. So please welcome Dr. Wolpe and take it away.
So the first question is, what ethics is about is how we work out conflicts and values. And as you'll see, all of these questions are issues of conflicts of values. And all of these extraordinary talks we've just heard about the clinical issues in space travel, some of them raise profound ethical issues, some of them less so. Then secondly, I'm going to focus primarily on commercial space travel. I worked at NASA for a long time, and NASA had come to certain kinds of conclusions about many of these issues. But as we move to private space travel and commercial space travel, these issues will come up again because the answers very well may be different. Commercial space travel is not bound by the precedents set at NASA. And so we need to rethink these issues, and we may end up having multiple answers to some of the way some of these issues are handled in the commercial space. Third, when we talk about fertility and reproduction in space, we're talking about an enormous range of possibilities, from a male that simply goes up and comes back down for 6 or a female for that matter, for 6 or 9 days, to a permanent settlement on Mars where people spend their life course there, where reproduction happens in a much lower gravity field. So we're not just talking about one set of ethical issues. It depends a lot on context when we talk about things like reproduction, gestation. And then finally, as a preliminary question, I don't think that the role of the ethicist is to provide the ethical answer, except in certain very specific case. I think it is to frame, define, and show the parameters of questions. And then especially in something like space travel, there has to be consensus groups that come together and come up with answers. So I'm not going to be giving you what I think are the answers to these questions so much. I'm just going to try to raise them. All right, so of course, the first set of questions are around health risks, as we've all heard. With males, spermatogenesis is certainly one of the big issues there. I mean, the other issues around cancers of susceptible body parts, thyroid cancers, things aren't as much about fertility and reproduction. So I'm going to focus primarily on that. So, the question, there are other things that we do terrestrially that could affect fertility in males. We have certain protective measures we put in place. We allow, in some cases, males to make the decision to put their fertility at risk. Probably the same thing is going to happen in space, where a good informed consent will then allow a male to make decisions about storing, cryopreserving sperm, and perhaps putting his fertility at risk by going into space for long periods of time. Age of consent might be an issue that we would consider. What's going to happen when we begin to take regular trips into space, when they become less about, people of extraordinary high worth going into space, but they become more of a routine kind of travel. Female fertility, same thing. We've heard so much about the kinds of dangers that they pose to women's fertility. And so for both men and women, informed consent of all of the risks, possible long-term effects, are going to be important. As we'll see in a minute, so is much better research so that we can define those risks much more accurately than we can now. Then we move into questions of things like conception. despite the mouse studies and other non-human studies, we really don't understand what human conception is going to look like in space, what the effects of radiation and microgravity might be on conception of, increased risks of central nervous system development problems, of genetic mutations. So all of those are issues that are going to have to be worked out, I think, before we can ethically engage in conception and childbirth and gestation in space. Another interesting question is, what will be the role of a partner in consent of those kinds of issues? Right now, that is a contested issue here on Earth. What's going to happen if a pregnant female wants to go on a long duration space flight? Will we allow that person's partner, assuming that there is a male partner with whom they are that's identified and attached in some way, will that partner have any rights in terms of consent? These are questions that we're going to have to ask moving forward, not only in terms of things like space tourists, but what about a woman who's employed by a space company to be a pilot or some kind of an attendant, or whose employment requires regular space travel? How will we think about conception, fertility in a person like that? Then the issues of gestation are part of that as well. The risks of radiation in a long duration space flight over the course of gestation. Again, all of this requires much deeper and more specific knowledge of what the potential impacts are, what the probabilities are of various kinds of susceptibilities, which all speaks to the need for research, and we'll get to research in a minute. At a place like NASA or the space agencies, the astronauts who went up were tested for all kinds of physical and psychiatric health measures. When we talk about regular space tourists, when we talk about the fact that in a commercial space flight, you can have different kinds of roles on a craft. You can have the employees of the space company that fly and run the craft. You can have space tourists that pay a lot of money just to go up for the recreational value. You can have commercial customers who want to go into microgravity, perhaps to test a product. You might have pharmaceutical companies that want to send someone up into space who then takes a substance to see what the metabolic impact of microgravity is. So you have very different, you might have government officials. So you have very different actors on that spacecraft, unlike at space agencies when they were all astronauts, even if their roles were different. So then we have to begin to ask, what kind of screening are we going to do? Are we going to allow people up with comorbidities? Remember, at NASA, this was tested very extensively. But if I'm a space tourist or I'm going up as a commercial customer, what kind of testing is the commercial space agency going to ask for a space traveler who might have either physical or psychiatric comorbidities? Also, psychiatric concerns in general. Will there be screening for mental health before people go into space? And that includes general mental health and mental health around issues of reproduction and fertility. What about alcoholism and drug use in space? Are these going to be things that commercial space companies are going to be really meticulous about, given that they are based on consumer payment for the right to fly? Will they then begin to compromise these kinds of standards in a way that space agencies did not? We know that stress, isolation affect gestation, affect women's reproductive careers. Are those also going to be issues that we think about in terms of women's gestation in space? Sleep disruptions, a very well-known part of being in microgravity. Astronauts have a lot of trouble sleeping. Diet and nutrition limitations. These are, excuse me. These are also things that we have to think about in terms of the whole package of any long-term space platform in which there might be reproductive activity. And then finally, exercise. We know how important exercise is in space. Astronauts lose muscle mass and bone mass. Exercise is absolutely crucial just to maintain it to the degree that's possible. Are we going to make it mandatory that people exercise in space? If not, that muscle and bone loss could itself have impacts on reproduction. And then finally, or perhaps underlying all of this, is the ultimate question. How are we going to balance individual autonomy to take risks against the responsibility of a space company to protect the people that use its vehicle to go into space? At NASA, this was a very explicit set of questions that we talked about a lot in the Chief Health and Medical Office. How do we do that balance? How do we make sure astronauts are both protected while at the same time maximizing their ability to go into space? But in commercial space travel, there isn't the same kind of oversight. We don't know how these companies are going to think of balancing an individual's right to take these kinds of risks versus the sort of fiduciary responsibility of a commercial space agency to protect the people in its using its vehicles. So those are some examples of the health risks problems. Now let's move to the question of our responsibility to the fetus and to the IVF was brought up, embryo selection, looking downstream to when we actually have settlements on the moon or Mars. Are we going to allow embryo selection? Are we going to allow genetic engineering? If, for example, we got to the point where we had really clear genetic markers for susceptibility to radiation-sensitive cancers like thyroid cancer, would it be ethical to choose either space travelers or embryos that showed a genetic marker of resistance to a radiation-sensitive cancer? Those are questions we're going to have to answer as we move forward. They're not short-term questions because they're not important in the next decade or 2 or 3, but they will be downstream questions that we should begin to talk about now. As people have mentioned, a developing fetus almost certainly will have susceptibility to radiation, to microgravity, to other stressors of space travel. What is our responsibility to them? We've already seen in mice certain central nervous system abnormalities and microgravity, the increased genetic mutations. Do we have the right as a society to say, until we can find acceptable ways to mitigate those risks, we will not as a society allow pregnant women to go on long duration space flights? That's a question for the society as a whole to answer. When you talk about birth in space, of course, you're talking about a whole other set of kinds of problems. If you think of birth in microgravity and you think of, or miscarriage in microgravity, which involves, significant bodily fluids, that can, how is that going to be contained in a space environment? What if you have a birth outcome where the child has special needs, abnormalities, heart defects, things like that, in a low-resourced environment where you don't have the ability to rush them to a surgical theater with sophisticated equipment? What level of risk are we willing to take in birth in space travel? And then we have the question of postnatal development after the child's born. What do we owe to that child in terms of their development? Do we, we will already know that in microgravity, they are going to have certain bone and muscle deficiencies. Excuse me. We don't know how long certain kinds of problems might take to show up. I'm sorry. Hold on one sec. Sorry. And so what do we owe to that child in terms of their health and in terms of their rearing? Imagine rearing a child on Mars in that reduced gravity who will never thereby be able to return to Earth's gravity. Their body does not develop enough bone and muscle strength to exist in Earth's gravity. Do we have the right to say you don't have the right to restrict the child's life to Mars by rearing a child in a lower gravity environment? Then there are the issues of resources, both on a craft, on a settlement. Craft have reduced life support resources, water, oxygen, space. Crafts are toxic environments. There are toxic chemicals in HVAC systems and other places. And we don't really know what a craft is going to look like that's commercially developed. You know, these things were studied very carefully in NASA spacecraft. And to what degree will we create radiation shielding? You know, radiation exposure, its impact is a function of multiple variables, of the time of exposure, of the age of the subject, of the types of cells we're talking about. You know, certain cells in the body are more susceptible, rapidly multiplying cells, gametes, and the efficiency of shielding. What kind of new shielding might we develop that is more protective, either in a craft or perhaps body shielding that can be worn? Medical resources, if you have an emergency, of course, spacecrafts are limited in the kinds of emergencies that they can cope with. And what about a newborn? What about incubators and extracorporeal oxygenation and surgical abilities? And then, of course, there's a question of limited nutrition potentially for a newborn in a space-based environment. So these are all questions about our responsibility to children postnatally and what kinds of things we are willing to risk and not risk in their development. One of the things that NASA could control easily, because the space agencies in general, not just NASA, had a lock on human bodies in space, and therefore they had a monopoly on research on human bodies in space. Well, now space companies have a very rare resource, these commercial companies, which is human bodies in space that are potential subjects of research. So how are they going to protect those human subjects from what could be damaging or problematic or harmful research? The common rule suggests that any human research subjects, of course, this is dependent on having gotten federal money. Let's assume for a moment that the commercial research companies do get some federal money. Then those subjects are come under the common rule, which allows them to withdraw at any time, to refuse to participate without penalty. Well, if you're a commercial company who has contracts to test human bodies in space, and a customer is not interested or willing to do that, is it a violation of the common rule for you to refuse them passage? That's a question we have to figure out. If a company doesn't accept any federal funds, then they have every right to tell you that you cannot fly if you are not willing to subject your body to human subjects research, which is a coercive situation to put someone in who really wants to engage in space flight. So we have a potentially problematic problem that we have to solve when commercial companies become monopolies around human subjects research. There are no regulations about these things that are applied, or at least we don't know which apply and which don't. It's a gray area that's going to have to be defined. And as I said before, remember, we have different kinds of travelers on those spacecraft. We have employees, we have tourists, we have commercial customers. Will there be contractual relationships about human subjects research? If a pharmaceutical company wants to test a drug in microgravity, will it be able to contract with a commercial space company whose job it is to get them subjects? And what kinds of abuses or problems might happen when a commercial company, for its own financial benefit, needs to recruit people to put their bodies at risk in space for a pharmaceutical test? What happens to sort of consent free of coercion? We need fully informed consent. We need individual autonomy around human subject research in space. And we also probably need some kind of co-design. This is something NASA learned at the beginning when astronauts were pretty much excluded from scientific review and ethical review of the research that was going on. And finally, after a series of issues and problems not worth going into here, it was recognized that including astronauts on the scientific review committees, including astronauts on the ethics review, gave them more buy-in to these kinds of processes, and maybe the same thing will end up being true with commercial passengers as well. And then finally, commercial interests themselves. You know, one of the things you're supposed to do in human subjects research is determine the balance between the risk of the experiment and the benefit to be gained. What is the benefit of commercial spaceflight? Is it only financial, or is it also sort of learning about the human body in space so that future people can travel in space? Or is it perhaps trying to make sure that when the asteroid hits and Earth becomes uninhabitable, we have populated other planets? How do you think of the idea of benefit when you look at the risk of space-based research and try to balance and say whether the risk exceeds the benefits? We're going to have to come to some consensus of what benefit looks like. Should a commercial space company require pregnancy testing before flight? And if so, does that then give them the right to say, no, you can't fly because you are pregnant? Can you mandate contraception in long duration space flight? Should, you know, cryopreservation has already been discussed, and it was discussed at NASA. Can you mandate cryopreservation of gametes or embryos before commercial space flight? We don't know yet what the impact will be of government regulations on things like frozen embryos in space. Again, this is going to be many decades in the future, but these are also things that government regulation is going to have to think about. It's really important that commercial companies share information around these things and don't think of these issues as proprietary, because this is, even though these are private companies, this is a human shared endeavor. There aren't any standards right now. They have to be developed. And then also the final question for space companies is, who's going to get to fly in under what's the conditions, who's going to have the privilege to fly, what kinds of requirements and prerequisites will be required for someone to fly, and how does that impact things like equity and fair access? So finally, a quick set of recommendations. We need to establish an international framework for this. We need to implement precautionary policies. This isn't the time to take significant risks with people's health. This isn't an emergency. We have time to think these things through and do the right research. The emergency is only in commercial competitiveness, and that to me is not an ethically acceptable reason to put people at significant risk. We have to conduct the correct ground-based research prior to space-based research, because, you know, sending people into space to test things like gestation and pregnancy is itself ethically problematic. We have to make that research transparent and collaborative, and we have to protect the fertility and the future of fetuses and neonates and pregnant women, and even men's fertility. We have to think of them in a precautionary way. And then finally, we have to establish a collective industry industry ethics review board to think about these things carefully, thoroughly, and then to come up with standards that are not restrictive, punitive, or that keeps the industry from developing, but that also protects human life, that protects human subjects, and that makes space travel something that we can all endorse. Thanks for your attention.
Let's go to the Q&A, but I think we have a guest here, Paul, who'd like to
Yes.
introduce.
So quickly before the Q&A, I want to introduce Sarah Lynn Mark. She's a renowned leader in women's health. She worked with me at NASA, where she was really a pioneer in women's health and space. She's an endocrinologist, a geriatrician. She's worked with DHHS. She's worked with almost every federal agency on these kinds of issues. And she's the CEO of SolarMed Solutions, a company where I also work. Sarah Lynn?
Great. Thank you so much, Paul. And it's a pleasure to be part of this very, very important symposium. I think as we envision the future, we have to learn from the past. And I want to take you on a very, very quick journey towards the past. Perhaps nowhere more in medicine does reproductive medicine have the political, the psychosocial, and really the elements of the community than you have in reproductive healthcare, especially female reproductive healthcare. At the turn of this century, right before, we had at NASA an important meeting to discuss having an all-female flight. And during that meeting, of course, we discussed some of the issues that we're talking about here today, including contraception. But there was a discussion from some of our male astronauts who were talking about some of the issues that they were facing. Not only in regard to fertility, but also some of the family psychosocial issues. And they had been basically felt that they didn't have a platform to talk about it. So that stayed within my framework as we began to develop around 2000, 2001, the first decadal review to evaluate the impact of sex and gender and how the body adapts in space. Shortly after the decadal review was announced and published, we had some comments from our female astronauts that they were very concerned about some of the delays in the shuttle program. This was before even the Columbia accident, and they were concerned about their fertility. I was invited to a meeting, a high-level meeting, because of the decadal review. I was there. And some of the comments were that if female astronauts were concerned about their fertility, that they should just not be part of the program. They shouldn't fly. And of course, as we know, that's gender discrimination, especially since we have the technology to manage it. What was very interesting is that it was around the same time that we were hearing about, abnormal frog development, especially male frogs due to environmental hazards and phytoestrogens. Some of my colleagues at Yale were looking at capacitation and sperm and radiation impact. So I began discussing developing an infertility policy for NASA, an ART policy, but not just for female astronauts, but also for our male colleagues, for our male astronauts. Fortunately, it was accepted, but as I mentioned, the political environment certainly shaped decisions. And around August of 2001, President Bush announced the stem cell announcement about how he was going to use stem cells for research and that we could utilize cells that had already been developed, but not to develop new ones. So to get this policy through NASA, we were able to do it in regard to medical care. A lot of the scientists and clinicians that were part of our decadal review were offering to provide care at cost. We were able to get therapeutics, medications at cost as well, but we were not allowed to do any storage of gametes as well as storage of embryos. I am happy to say that this policy is still in place about 25 years later. It's serving both male and female astronauts. And I think it can be used as an example of how we can develop policies not only to meet the needs of one gender, but for everybody. And despite the political climate, it can survive. So I just want to provide that backdrop as we go forward, because some of these issues are going to be very critical, very important as we look at the commercial space sector. So thank you for your time.
No, thank you very much, and thanks for joining us. And I hope you'll join us for the after party afterwards. Can I pass the baton to Virginia? Perhaps you'd like to start the Q&A? Is that
Absolutely.
okay?
Absolutely. Thank you, Giles. So, Dr. Wolpe, I have a question for you. So, and it's from me. You mentioned that that it would be a good idea to have harmonious, comprehensive approaches to how we handle ethics for astronauts, regardless of what type of astronaut. How would you handle the different international and intercultural aspects when it comes to astronauts from all over the world?
Yeah. Well, there certainly are cultural differences in standards, but what's interesting in terms of human subjects research is we've seen over the last few decades a convergence of different groups. In fact, I went to Russia with NASA when I was there to the Russian aeronautic and space meetings, and we did what we thought was going to be a little ethics session, and hundreds of people came, much to everyone's shock. Why? Because at that moment they were talking about joining an international consortium. And in order to join that international consortium, they had to bring their human subjects into international standards. So you see, there can be a lot of pressure internationally for groups to conform to a set of standards. And finally, in commercial life, you often find that companies really want reasonable regulations, because what they don't want to have happen is to set up a system or set up some set of procedures that then become seen as unethical or problematic. So they would much rather know what the boundaries are, what the ceiling and floor is in which they can work. So I would think that, especially with something as sensitive as this, where bad PR could really sink a company, I think companies would probably be willing to collaborate to try to set international standards.
Thank you. Fathi?
Yes, thanks. I have a question for actually Dr. Mathyk as well as Dr. Jones and Dr. Starks. So actually, it's a question from one of the participants. Her name is Karima Jeroubi. She's originally from Algeria. And actually, the question has two elements. The first one is kind of related to the fact that the focus of your presentation was mainly on the effect of microgravity. Yeah, there were some elements related to hypergravity. But what about partial gravity, lunar gravity, Martian gravity? Any insights in relation to the impact of those partial gravity on the reproductive system from a male and female perspective? And then the second part of the question is related to the fact that how can we leverage whatever we learn from exposing different type of specimen in space to IVF on Earth? So in I guess in some aspects, what are maybe opportunities for what we learned from space exploration that could be beneficial for us on Earth?
Well, Fathi, I'll go ahead and take the first response. Certainly, the question of partial gravity effects on reproductive system is not completely characterized. That was one of the things we wanted to do on ISS when we had the CAM as a part of the program. As you know, the CAM was one of the early cuts to the program, the centrifuge accommodation module. But we wanted to look at those kind of things. How does 1/6 gravity or 1/3 gravity provide protection of normal physiologic systems? The speculation is that either lunar or Martian gravity would have a relatively normal physiology. However, the details of terms of cellular expression, as you know, are not completely known. In regards to the second part of the question, do you want to answer that about how we could use? Yeah.
So there was a mission that was called the experiment, Micro 11 experiment. I think it was sent up on SpaceX 14 in 2018 that actually did send up human cryopreserved bull and human sperm samples. And part of that experiment was then to thaw them in on the ISS and look at how those sperm took on motility and then took on potential flagellar movement. Unfortunately, none of the data are published. There is a little bit of a teaser on YouTube that I was looking through that did find that the sperm were actually able to undergo the cryo depreservation process and take on some flagellar movement. So I think it's a great question that we definitely need to start exploring by, potentially expanding these type of experiments, actually looking at human samples and the effects of either microgravity or, to your point, Dr. Jones, partial gravity, if we're thinking about habitation on lunar surfaces or Martian surfaces as well.
Thank you. Actually, I was involved with Micro 11. So as part of the previous I guess, medicine talk, I kind of gave a brief introduction to the project. But yeah, to the point, indeed, we have not yet published the data, unfortunately. But I mean, second part of the question was more related if there's anything that we can take away from space exploration that could benefit IVF on Earth. And I was wondering if you can shine on that as well.
I can go, or the male reproductive experts wants to go first.
Please go ahead, Begum.
Okay.
Go for it, Dr. Mathyk.
Yeah, so basically, it's like we always say things about like the unfavorable effects of different microgravity or radiation, but I think the fascinating part is pushing us to develop technology. So more automated systems and everything. And we have like the IVF sector developed like tremendously. Like the first baby was born with an automated ICSI. This is like one of the groundbreaking things happened in the IVF world and embryology world. So what we can benefit from space technology, I think creating the automated systems that is not going to rely on astronaut changing the culture. One is that. The second thing, it is going to push us and force us to come up with new cryoprotectants, like, for radiation exposure or like different temperature alterations. So that will definitely benefit the IVF on Earth. Maybe we may not need like what we currently have, the cryo tanks in the future. We may have different, versions, different temperature settings where we don't need the 196 anymore. Like, they're trying different methods like high osmolarity or freeze dry sperm, but we are not there yet on Earth. But that the pushing towards to space can actually benefit the Earth IVF and reproductive health as well. And the other thing is that about partial gravity, we have data on the impact of partial gravity on musculoskeletal and different organ systems, but it hasn't been investigated in detail for the female reproductive organs. So that's another thing. I think it's so difficult to say it affects or doesn't affect when you don't have the data. You can only speculate that because of the gravitational changes, even though it is a little bit or not the same, it may. But again, it's so difficult to make a definitive conclusive statement without having the scientific data.
Thank you very much, Dr. Mathyk, Dr. Jones, and Dr. Stocks. Greatly appreciate.
And Dr. Karouia, the real test will be when we have the first lunatic or the first Martian born on that surface to see if they look like terrestrial humans or they have their own appearance. That will be the acid test.
Mm-hmm.
Good point. All right, Jill, do we have time for another question for maybe Chris to ask?
Yes, if Chris wants to ask just one more question before we wrap up, we'll give him the chance, if that's okay.
One maybe question for Dr. Wolpe or for anyone, but some recent technologies for somatic CRISPR editing have been able to be a cure for beta thalassemia and sickle cell disease. Could you imagine this be a means by which you could enable an easier transit between the planets, or even someone who wouldn't normally be able to go to space suddenly enable them? And your thoughts on that, Dr. Wolpe, I think.
One can think of 100 terrestrial uses of genetic engineering before we start creating genetically altered astronauts. And we haven't even agreed on those yet. In fact, using genetic engineering to cure disease is still controversial. We have found over and over again that genetic intervention isn't simple. And what we thought would be a, single point effect of a genetic intervention has collateral effects that we didn't anticipate. And also our tools aren't completely accurate in terms of knowing what insertions and other things, what kinds of implications they'll have. So I think we need to solve the question of what is an appropriate use of genetic intervention first. I mean, I'm all for preventing disease before I'm, in favor of genetically altering the human genome so that people can go into space. And by the way, the final issue about that is it's a different question if I, as an adult, could use those technologies to alter my body to go into space. But if you're talking about creating embryos who can then grow up to be space travelers, you are messing with their genome
Oh, yeah.
in order to project that later they will have a role that you are imposing on them. That's deeply ethically problematic. It's no different than me deciding that I want to genetically engineer my son to be, 6'10" because I want him to be a basketball player.
Yes, but the FDA approval for the sickle cell treatment's already gone through. Do you think that's too far, the FDA approval?
No, I mean, I think sickle cell is a debilitating disease. It has to be done with extraordinary caution. Right now, the people that are doing this kind of work are academically based. There's enormous oversight. My worry is what's going to happen when it does go into the commercial sector and whether that kind of oversight is going to be as rigorous.
Chris, it's a really relevant question that you ask. When I was working with Brigadier General Pete Warden, when he was directing the military spaceplane project, he specifically asked the question, what can we do to enhance astronauts' capabilities to resist microgravity and radiation effects? And we even talked about splicing genes from radiodurans, one of the bacteria that's known radio resistant, into a human to enhance their radiation resistance properties. So there are people asking such questions. It's relevant, although I agree with Dr. Wolpe that the ethics are still not where we want them to be.
Yeah, there'd have to be like the key phrase is there an unmet medical need? If you're doing something, sporadically, of course, that's not really ethical. But if there's a need where the choice is, survival or not, that's a medical need. But it's short of that, everything else is pretty problematic. Yeah.
But I think
And by the way,
disease
I'm just going to say,
disease genomes versus healthy genomes. Which ones are okay to alter, and is it really okay to alter a normal genome?
And before we get to alteration, we're going to have to make the decision about testing. Are we going to begin to test embryos for things like susceptibilities to radiation, susceptible cancers, like I said, and other things, and then say, okay, these bodies, without manipulation, just in terms of their natural progression, are more fit to go into space than those bodies?
I think I would like to just say one thing that we do it daily basis in an OBGYN life, like shared decision making and autonomy. I think the more data we have, we still don't know. This is just like scratching the surface and the tip of the iceberg. So the more data you have, then you can consult them. What are the consequences of either doing something or not doing something? Then, we should have to have like consents, shared decision making, and respect the autonomy of the participant as well.
Okay.
All right.
Over
Well, I
to you,
think
Fathi.
it's time to wrap up. And so I just would like to thank everybody, and in particular, our three distinguished presenters. And so thanks again, Dr. Mathyk from the University of South Florida. Thanks again, Dr. Jones and Dr. Stocks from Baylor College of Medicine. And thanks again, Dr. Wolpe from Emory University, for those outstanding presentation. I think all the participants are very much grateful for hearing about you as experts in the field and getting insights in reproductive space medicine. Similarly, I would like to thank also all the moderators and panelists. And so thanks again, Professor Mason. Thanks again, Sir Wotring from the International Space University, and Chris from Weill Cornell University. Again, thanks for being there and helping in moderating the conversation. And then Giles, thank you also for putting that together. I think next, maybe we can have a few words from you, Chris.
Yes, I just want to say I really enjoy the broad international perspective. And I think this is really some of the papers we put in I put some of the speakers' papers in the chat and some of our papers from collaborators, some of our lab. There's a wealth of literature that's come up during a discussion. And I think this really is a second space age where we have, obviously commercial entities, but new sovereign activity. We have a lot of people looking with their eyes out to the skies. It's really extraordinary time. But I think having great universal informed consent has come up in the discussion here, and enabling people to really know the risks is important, but then being able to I think as Dr. Wolpe said, ways where people can share the data and have international collaboration helps us all learn faster. So I think it's really an exciting time, and really enjoyed the discussion, the debate, the back and forth, and look forward to seeing you all in the coming years and decades, hopefully for many research and big questions.
Thank you. And Virginia, a few last words?
Thank you, Giles. So yeah, fascinating discussion, and we barely scratched the surface of all the questions that came up. There's a lot to talk about. So I just wanted to point out to everyone that I'm with International Space University. We're a tiny little university in Strasbourg, France, that teaches only about space. So if this topic was interesting to you tonight, please check us out. I've just posted our web address in the chat. We're easy to find. You can get a master's degree with us. You can do a professional development program that lasts just a couple days or all summer long. Take a look and see if you want to learn some more about space with us.
Okay, well, thank you very much. And as part of I3, and I'd like to thank all my team on the I3 initiative, which is Jacques Cohen, Peter Nage, Marianne Chevetes, Thomas Elliott, Fran Farley, Violetta Sora. And join us next week when we'll have a webinar called Unstoppable, all about the advancement in technology in the IVF laboratory. So again, thank you very much for joining, and I'll see you again soon. Thank you, everybody. Bye-bye.